And so we'll be going back to the synthesis of fats. We said that acetyl-CoA is the starting point for the synthesis of fats, but what is the committed step? What does acetyl-CoA have to be converted into to go down a synthetic route? Malonyl-CoA. And so this happens in the cytosol. So if that is the committed step, then the regulation is going to be controlled at that enzyme. All right. Now, we have lots and lots of different types of regulation. Okay. Mainly we talk about covalent modification, and then we talk about allosteric. All right. Covalent modification is usually stemming from a hormonal response, a hormone sent out in the body in response to a condition, and usually you get a covalent modification. Mainly, we've seen phosphorylations, dephosphorylations. That happens quite often, although there are others. There are methylations, you might put an adenylate on, but we've seen a lot of phosphorylations. The secondary type of regulation is an allosteric, where binding occurs on the enzyme, not at the active site but somewhere else, that modulates. Either it can enhance or it can inhibit the activity of a protein. These things work in tandem, because you could be sending a signal out hormonally that says go, 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 but the particular cell that you're in could have concentrations that really don't need it to go, and so you can have two competing signals that adapt the activity of the enzyme to fit that particular cell at that particular time. All right, we talk about three signaling molecules: insulin, glucagon, and epinephrine. What does insulin signal? You're full. You've just eaten. You've got lots of molecules to make energy, so you have lots of starting material, and usually you have lots of ATP, well-fed state. What is glucagon? Starvation. What's epinephrine? Well, it's going to, you're going to force energy to be derived from the system. Doesn't matter if you're well-fed or if you're starving, because the lion is chasing you, and this is the fight or flight, okay? Or the pop quiz got in front of you. Epinephrine comes out then too, right? Fight or flight. Now, one other thing I don't know if I've actually mentioned to you before, insulin will affect both liver and muscle, okay? Those are our two main tissue types that we think about when we talk about metabolism. Glucagon, the starvation state, affects liver primarily. It doesn't affect muscle. That particular hormone, there's no receptor for glucagon in muscle. Epinephrine, in a lot of ways, will reinforce glucagon's signaling pathway. Epinephrine does actually hit the muscle, and so this is one of the reasons why we talk about glucagon and epinephrine together. All right, so the enzyme is the acetyl-CoA carboxylase. That is the one that makes malonyl-CoA. I have two different versions. The inactive monomer, okay? So the protein is by itself, it's phosphorylated, dead activity, okay? The active polymer that I'm going to do reactions and put a CO2 onto acetyl-CoA is a polymer that binds together, and it's dephosphorylated. Okay, so those are our two states. In insulin, when the cell accepts insulin, it activates a protein phosphatase, usually called PP, that's the protein phosphatase, and the version is 2A. So phosphatase takes off a phosphate. So what it will do primarily is take all of these monomers, take off the phosphate, and shift it into the active polymer. So insulin makes this go, which then allows us to build fat. Okay, why in the world do we do that? If we have lots of energy molecules and potentially a lot of ATP, this is the time to store. And so that's what insulin will do, and it will make the fat cells. Primarily, the fat is being produced in the liver so that it can be repackaged and sent to the adipose tissue. All right. When I look at glucagon and epinephrine, we're going to have a different route. What's going to happen is we're going to stimulate phosphorylation. How we do that is we upregulate a AMP-activated protein kinase. And so if I have a polymer that is active, the kinase will phosphorylate them. They will dissociate and become inactive. Both epinephrine and glucagon will cause the shift between inactive and active. All right? Now, there's a whole lot of other things that go on that are allosteric. If I look at citrate, if it becomes high in concentration, all right, citrate is the first step in the citric acid cycle, and if the citric acid cycle is backing up, okay, that means I have fulfilled lots and lots of molecules going to be broken down to make ATP. If citrate starts to back up, that means I've overwhelmed the ATP synthesis process, and so citrate builds in concentration. That means if I send acetyl-CoA to citrate to go that direction, I'm going to waste the molecules. This is an indicator I have made as much ATP, or I'm at the maximal rate of making ATP. Instead of sending the acetyl-CoA into the citric acid cycle, send it to fat synthesis. Well, how do I do that? Even if hormonally it says don't go, if this concentration goes high enough, it will cause polymerization. All right? The phosphates say stop, the citrates say go, you get a little bit of activity. And so you start sending the CoA, the acetyl-CoA, to synthesis of fat. Another feedback inhibition or mechanism is the actual end product. Okay? If the end product becomes high in concentration in the cell, I've made lots of fat. I can't get it out of the cell fast enough to get it to the adipose tissue. What's going to come back is the end product will bind to the committed stepped enzyme and put it into the inactive form. Okay? And so if I'm making lots of fat, I can stop it or slow it down, although citrate can also make it go. Okay? But if I do not have fat and I do have a lot of citrate, I can make this polymer go very, very quickly. And it's actually almost as active as the phosphorylated state. And so the end product turns it off, citrate turns it on, and then if it's phosphorylated, it's off, and if it's dephosphorylated, it goes. One more item is AMP itself. Okay? AMP, if that concentration goes up, what we're going to do is stimulate the AMP kinase. It was seen in that first slide, but the AMP actually binds to the regulatory portion of this kinase, and it will cause the phosphorylation and the deactivation of that particular enzyme by making them monomers. AMP also could be a signal that there's low energy in the system. If there's low energy in the system, synthesizing is not likely to happen. And so either I can stimulate AMP to be created by a hormonal response, or if just AMP starts building up in concentration, it can signal the enzyme to stop itself. All right. So I've made lots and lots of long-chained saturated fats, but we've only made it up to 16. At this point in time, if I want to make any fat that is longer than 16, the 16 carbon intermediate is transported to the cytosolic face of the ER. There's a whole set of enzymes that will elongate it past. They are very similar to the elongation steps that we saw with the normal synthesis. They also use malonyl-CoA as the activated intermediate, and it will elongate to longer fats. The other thing that we can do at the face of the ER is use molecular oxygen and some electrons and cause an unsaturation. What will happen in the steps is, for the unsaturation, the middle of the reaction, we will get a hydroxide containing intermediate, and then we'll have an elimination. So it's a stepwise process, and that will pick up a proton. The oxygen reacts with the straight chain that is saturated. It will create the hydroxide, and then we can eliminate. But to do that, it has to have extra electrons. Those electrons come from NADH, transferred to FADH2, which is transferred to a cytochrome, and cytochrome carries electrons by the iron center, and then that iron center will give it to the enzyme that will facilitate the addition of the hydroxide, which then facilitates the elimination. And so molecular oxygen and electron pairs are needed. The mechanisms that occur with these kinds of reactions use an iron as part of the active site and gets into a really strange oxidation state. A little complex for what I want you to know, other than the fact that we need molecular oxygen and electrons to create unsaturations. And normally they are created in the cis form. Now, we can unsaturate between any carbons numbered 1 through 9 in our bodies. If you need unsaturations past 9, the only way that that can be done is in plants. Plants have enzymes to add unsaturations past the 9th. Now, we can work on free fats and actually place the unsaturation in them. The plants actually do their unsaturation past the 9th when the fat has already been incorporated into a membrane lipid. And so the enzymes are optimized for membrane lipids, and they can add the unsaturations that go very far out. What we get here are fats that are essential. Okay, so you have to eat a fatty plant to get these, and some of the things that we've seen in the past were hormone-like structures called the eicosanoids that come from arachidonic acid that have unsaturations past the 9th. It creates a whole class of prostaglandins, thromboxanes, leukotrienes, and these are important molecules. They must be eaten to be incorporated into our system so that we can use them in as regulatory molecules. All right. I just don't think too often about fatty plants. Just don't think of a plant as being fat, but there are quite a number of plants that actually give us a lot of fat. One that becomes very, very popular that in the past couple of years, avocados. You can get lots of good fats from that. Unfortunately, there are some disruptions in the supply chain that have occurred recently, and avocados are becoming much, much more expensive. And the American diet has really tried to, or has really found the avocado to be highly prized. So avocado prices are going up, which really messes with my guacamole making that my wife does. And I'm just kind of upset about it, but we'll get past that. All right, so we can make the actual fats, the free fats. Free fats don't like to be in water, okay? So the fats are incorporated into molecules to be stored, and we put 3 fats on a glycerol, we make a storage fat, or they are incorporated into membranes, and so we're going to add them to molecules to be incorporated in the membranes. Most of the time, the glycerol that is used for fat is derived from glycotic intermediates. And so dihydroxyacetone or glycerol can be reduced and or phosphorylated into the glycerol 3-phosphate, which is the basis for which we build both the triacylglycerols and the phospholipids. In this case, to create the triacylglycerol, the fat storage molecule, pretty simple. We take the fatty acid that's attached to CoA, it gets brought together, and the hydroxide will attack at the carbonyl, create an ester. CoA is the leaving group. And we do that again, and CoA is the leaving group. The last one, we dephosphorylate the third position, and again, the hydroxide attacks at the carbonyl to create an ester, and the CoA is a leaving group. We have the fat molecule, which then can be incorporated in adipose tissue. Most of the time, the fats are carried by albumin from the liver and incorporated into the adipose tissue, and synthesis can occur there, or the synthesis can be in liver, and it creates the fat and it incorporates into a particle that is exported to be delivered to the adipose tissue. The membrane phospholipid synthesis occurs in almost all cells, and the strategy is, one, create the backbone, one of which is glycerol that we just saw, add fats through either an ester or an amide linkage, and then attach a polar head group to that molecule so that you have a polar side and a hydrophobic side that can be incorporated into the membrane. A lot of times the polar head group then gets functionalized, or it gets a derivative made from it. What happens in the synthesis is we need to power the addition of the polar head group, and we use, in this case, the energy molecule CTP. Okay, we haven't seen CTP used very often to power something. Almost all the time it's ATP. Okay? Every so often we see a reaction powered by GTP. Here is one that is powered by CTP. What we're going to do is react the growing membrane lipid with CTP to get an intermediate that has a diphosphate CTP, or CDP, diphosphate. Okay. Now, there are two strategies, and the human body uses both strategies to create lipids. One, you can put the activating group on the growing phospholipid. Then the polar head group will come in and power or be powered by the leaving group of CMP. The secondary process is you can take the polar head group and put the CDP on it, activate that, and then allow diacylglycerol to attack it, and CMP is the leaving group. The outcome is the same in terms of what you get, but we have two different classes of mechanisms to actually put together the lipid portion to the polar head group. So strategy 1 and strategy 2. An example, or a couple examples of membranes that are created, membrane lipids. We follow the same synthesis that we see in fat synthesis up until we get to the point that we have two fats and the phosphate. Okay? If I wanted to make the true fat molecule, I'll dephosphorylate. But if I'm going to make the phospholipids, what I'll do is I'll take this phosphate right here and I'll react it with CTP. It will attack, and I will get two PIs falling off, and what I will be left with is one phosphate that was original, one phosphate that was on the CTP, and so it's kind of written weird. You've got a phosphate here and a CMP CMP, one phosphate plus another phosphate, that's the CDP, the diphosphate. In this case, I'm activated and I can bring in here the polar head group serine, or on this case, the polar head group inositol. Both of them are alcohols. And one of the hydroxides on either one of these entities will attack at the phosphate, and the CMP will be the leaving group. So I get phosphatidylserine or phosphatidyl'inositol. Both of these are found quite often in membranes. In the case of phosphatidylserine, it can be decarboxylated where the CO2 falls off and I get phosphatidyl ethanolamine, which is another another phospholipid that is found quite often in membranes. The inositol quite often gets phosphorylated in the 4th and the 5th position, and it sits in a membrane. We have seen this particular molecule before, okay? And this particular molecule is worked on by an enzyme called PLC. Do you know what PLC stands for? That's the worst thing about science. We have too many acronyms. Do you remember what PLC stands for? Nope. Looks like you don't. Okay, so it is phospholipase C. Okay, does that ring a bell at all? Okay, well, lipases are those molecules that break down fats. Phospholipase, well, this is a phosphorylated fat. And C is the position by which it breaks the bond. There are 4 phospholipases: phospholipase A1, phospholipase A2. I break the first fat, I break the second fat off. Phospholipase C breaks in front of the phosphate and the phosphate falls off. Phospholipase D breaks on the other side of the phosphate and the polar head group falls off. Okay? So if I use phospholipase C on this particular molecule, I make 2 entities, DAG plus IP3. What does DAG stand for? Diacylglycerol. And IP3 is inositol 3-phosphate. It's phosphorylated in the 1, 4, and 5 position. Okay? Now, this will upregulate in a regulatory molecule, signal transduction, one particular enzyme. Can you remember which enzyme it is? Not pKa, but it is a PK, pKc. The DAG will float over the pKc, make it higher affinity for calcium. IP3 goes to the ER, opens a channel and allows calcium goes to protein kinase C, and if DAG and calcium bind, the protein kinase C is activated and it will phosphorylate for a cellular event. Okay, that was seen when we looked at signal transduction. All right, most lipids the fats are held together to the base molecule through an ester bond. There is a couple of examples of ether lipids, and if you take a look at this, the synthesis is astoundingly complex, but they are there. They're not exactly sure why this particular bond is important for these lipids, but we do have a few examples. I do not want you to know the synthetic pathway of an ether lipid, just that they do exist. On the other hand, there is another base molecule which we add fats and polar head groups to, and those are the lipids that have sphingosine as the base, and we call them sphingolipids. In this case, we've got 4 major stages in its synthesis. We take a fatty acid and condense it with serine. So the base molecule that we have is serine, and we add the first fat directly to the serine. Then we add a secondary fat to make two fats through the amide. We have to desaturate it and then add a polar head group. And so here is serine. Oops, I didn't want to do that. Here is serine and the palmitate-CoA, and the reaction occurs with CO2 leaving. So CO2 will leave. I'll have a negative pair of electrons that will attack, kick up, kick down, CoA leaves. So here's serine attached to the fat. This is one of our membrane fats. All right. Second stage, NADPH reduces the ketone to an alcohol. What we really want to do is eventually get a unsaturation to occur. Once we have this alcohol here, we will add to this nitrogen another fat. And so I now have 1, 2 fats. That's what we need for every membrane lipid. All right. I will then desaturate, or water will leave, and I'll create a double bond. Oh, no, sorry. I will create a double bond in a different way. I will just actually create it with the use of FAD. And at this point in time, we have the base ceramide that can then have polar head groups added to it. Even though the structure looks very different from glycerol, they actually three-dimensionally are very, very similar. And so the way we draw molecules in a drawing program can make you think that they have a much different structure. Now, I've made a bunch of membranes, lipids. Well, how do I get them around? Well, it's happening at the ER. And what happens is we actually have vesicles that are transported. Now, most of the time when we're looking at a animation like this, what we're concerned about is what's inside. And so we may move around things inside the lipids, and we can exocytose something. But as those lipids move from one compartment to the next, you're also going to take lipids that were synthesized in one particular area, and it'll fuse to the next membrane, and I actually move the lipids from one membrane to the next. And so from this case, you can see from the ER, membrane lipids are moved to the Golgi, and then from the Golgi, membrane lipids are also moved to the plasma membrane along with whatever was inside. And so it's hard to get these membranes moved around because they're very hydrophobic. And so these vesicles that go from place to place can actually be a mechanism by which we move those lipids. All right. So we've made straight fats. We made membrane fats. We're now going to make other fats in the system, one of which is cholesterol. Again, the precursor is acetyl-CoA, and we're going to make a cholesterol from that. Now, acetyl-CoA has 2 carbons, and cholesterol has 27 carbons, so how many acetyl-CoAs do you need? Well, 2 and 27 don't match up. And so the answer is we need 15 of them because we're going to create an intermediate that has 30 carbons, and then we lose a few carbons to get down to cholesterol. Cholesterol is actually a very intensive, energetic process that we have to invest a lot of energy in, but we can synthesize cholesterol. The overview is to take lots of acetyl groups, put them together, okay? And we will make, eventually, mevalonate. Now that stage 1 that you see, we've already learned. This is part of the ketone body synthesis, okay? So stage 1, we've talked about it, and the liver can send out the ketone bodies to power the heart, okay? But if I want to make cholesterol, I will hijack that molecule and put it into another compartment to synthesize cholesterol. And I will make an intermediate that is 5 carbons long. Okay? An isopentenyl diphosphate. Now, there is a resonance structure that will work, and so there are two 5-carbon intermediates that will work together to do the synthesis. Now, we are synthesizing cholesterol, but we can also synthesize lots of other hydrophobic molecules, including the chlorophylls, the vitamins, and the quinones. Okay? In this case, which of those three can we synthesize? I mean, which of those can humans synthesize? Of these ones right here. Quinones? Yes. Anything else? Quinones are the electron carriers, so we have to have electron carriers. Can we synthesize chlorophyll? Why can't we synthesize chlorophyll? Because plants are the only ones that have them. Plants have the enzymes for that. Okay, that's cool. Can we make vitamin A, E, or K? They're essential. They are vitamins. That means we have to eat them. They are also synthesized in plants. But the same intermediate is used as the precursor for the synthesis. Plants will use those in their particular enzymes. We will condense the molecules together to finally get to a molecule that is 30 carbons long, called squalene, which will then convert into cholesterol. Okay. In reviewing a piece of the ketone body synthesis, okay, we get to this molecule right here, which can be sent out in the bloodstream if the CoA is taken off of it. And so we'll add lots and lots of CoAs together, and if by chance you want it to go and be used as energy, this molecule can be put into the mitochondria inside the muscle cell and then be broken down and be used as energy. If by chance you want to use it as a synthetic pathway, especially in the liver, it'll go into the cytosol and be converted into mevalonate. Mevalonate is the committed step to the synthesis of cholesterol. To do that, it's kind of interesting. It takes two NADPHs. What you're going to have is the first NADPH you will have the hydride attack, electrons kick up, electrons kick down, the CoA falls off. Now what is at the end is a double bond and an aldehyde. The CoA has fallen off. You use a second NADPH, you attack, you pick up a proton, and you create the hydroxide that you see there. Once you've invested the energy of two NADPHs, the mevalonate can only be sent to synthesize the hydrophobic molecules, mainly cholesterol, but we can do the quinones also. This is 6 carbons long. Okay, we are going to lose a carbon in this process towards synthesis. Okay, a little bit of an overview of what happens. I'll actually start showing some mechanisms in just a moment. We've got mevalonate, and we're going to use, watch this, 1, 2, 3 ATPs, all right, to create the 5-carbon intermediate. And then you can see, once we have that 5-carbon intermediate, we have a resonance structure that distributes it between two different resonance forms. Now, we haven't actually made cholesterol at that point in time, but we've invested a lot of ATP to get up here. We've also invested a lot of NADPH, which was derived from sugar, and so we're just throwing lots of energy into this molecule. All right. Once we get up to the third use of phosphates, okay, I said I used 3. We use one reaction to put that one on, the second ATP to put this one on, and the third ADP to put that one on. Okay? So 1, 2, and 3. Once we've got there, what happens is I'm going to decarboxylate. The electrons will form the double bond of the CO2. This pair of electrons will form the unsaturation, and it can only do that because I have a good leaving group of a phosphate. I get to the 3-isopentenyl pyrophosphate. That then allows us to make a resonance structure, put another arrow back and forth, to distribute between the two 5-carbon intermediates that we're going to use in just a moment. All right. To do the first reaction, we use the dimethyl allyl, and it does something very, very strange. We've seen it before, but this doesn't seem right. The phosphate falls off to create a carbocation. There is 2 reasons why this is stable. You break down that diphosphate coming off. So I release energy. The second is this particular carbocation can resonate, and the positive charge can be either in that position or on the methylene. Being right here is much, much more stable. Okay, being on a carbocation on a tertiary carbon is much more stable. And so, because I get energy derived from breaking down the pyrophosphate and the resonance structure, this is stable enough for the reaction to proceed. This creates a very reactive species that will react with the second 5-carbon intermediate that's in the other form. Now this is kind of weird too. Watch this. What's going to happen is this pair of electrons can actually attack and create a bond. And so the attacking bond creates that one right there. But when the electron pair attacks that positive charge, it creates a carbocation. And the carbocation in the tertiary is much more stable than the carbocation that is there. Okay? And so that is why that can proceed. Now this intermediate doesn't stay around for very long because that's still high in energy. To finish the high energy process, the last step is there is a base that will take this proton and create a double bond. And we've now created a stable molecule with no carbocations. And I went from 2 5 intermediates to one that is 10 long. All right. This geranyl pyrophosphate, if by chance I took this thing off and I put a proton on, looks just like the starting material before. Okay? So what can happen is I can do exactly the same mechanism in which that falls out, another 5-carbon intermediate reacts, and I will get a molecule that is called farnesyl, which is 15 carbons long. So I had 5, added 5, got 10. Had 10, add 5, get 15. I got to get up to 30. So what's the next step in this synthesis? What? Do it again, go to 20. You would think that is true. Okay? It goes 5 plus 5 is 10, 10 plus 5 is 15, and then it takes 2 15s and puts them together to make 30. Squalene. Now squalene, right then and there, can you see cholesterol? Does that look like cholesterol? That really doesn't look like cholesterol at all, okay? But that is the intermediate for cholesterol. All right. Did any of you use ChemSketch, inorganic? All right. A lot of times you draw a molecule and it just doesn't look right. But there's this little thing where you can highlight it and hit fix and it makes it look right. Okay. Now the squalene, you know, has a lot of flexibility because there's a lot of single bonds in there. What happens is the squalene will bind to the next enzyme just like this. Can you see cholesterol now? Can you see where the 6-membered rings are? And so even though it doesn't look like cholesterol, but when it fits inside the enzyme, it is just right there. Now, 23 reactions. We'll start that on Monday. All right. So at the end, you guys are working on your organic review quizzes. All right, so get that done and then start looking at the quiz about the lipids. Looking at a Moodle, I put a couple things up since last I saw you. We have the organic mechanism review Dropbox. And so you can either take a picture of it, convert it into PDF, or go to a copier anywhere on campus and send it to yourself. You feed it through, it'll scan the document, put it into PDF, and then you can upload it. So I can go ahead and grade that. It is due on Monday at 11:55. So you'll have until Monday evening to deal with that. And then I want to start you on the next quiz, which is about lipids. And so I want to talk you through what we're going to do here. All right, so there are 10 questions on this quiz. You have to answer 7 of them. All right, you must do number 3 and number 7 and then any 5 additional. And so you're going to have a question with multiple choice answers. A little tiny bit of the points you can earn is the actual letter. The most you can earn is the short answer about why the item you selected was correct. And then additionally, what I want you to do is give me a reference about where you found that information that you are using to base your answer. And so for each question, 0.25 is the letter choice, 2 points is the short answer and explaining why it's correct, and 0.25 is the explanation of why that answer is correct. And so what this is going to do, we're talking about synthesis of fats, we're talking about distribution. What we're going to look at is more clinical applications. And so we're going to give you a scenario about what you might see in a real case that relates to the material that we have. And so our material is very, very straightforward. We think about it linearly. We think about a synthetic process. But really what you're going to deal with is what happens when things go wrong. And so the process isn't working correctly, which then causes an imbalance. And where that imbalance happens and what builds up in concentration leads you back to where in the process something went wrong. And so this is the application of all of that you're studying, but what might happen and what you might observe. And so this is a application type of homework and activity. Now, when you think about application, that probably means it's going to be a little bit more involved. And so this is not something that you do in 20 minutes. You'll be looking up, and you'll probably need to look up resources to find this. But since they are tough, I'm not going to make you do every single one of them. If you get down a rabbit hole and you just can't find it, go to something else, okay, so that you can get the ones that you can find an answer to. And so this will be due, it says the 28th. So you'll probably be working in tandem, the organic portion and this as you're preparing.